But it's very nice to see all of you here. Actually, I just want to make a comment that this is the 9th year of the Myeloma Forum already. And if I look at the audience, there are many of you that were right here 9 years ago, right? So this actually tells us that myeloma patients are really surviving a long time now. Of course, we are not quite there yet because actually over this period of time, we have also lost some of our friends, but I think clearly we are making very important progress because if you look back at 10, 15 years ago, I wouldn't be in the situation where I'll be seeing so many of you that are still with us 9 years and, looking healthy and looking well and, go about your daily business. So this is very positive. And what I'm going to talk here for the next 15 to 20 minutes is further signal of progress, right? So, we are now moving beyond just the usual drug treatment, radiation, to, this whole new area of using our own immune cells to tackle the cancer, in this case, multiple myeloma. So actually, without much coordination, I think the talks have moved very nicely because Dr. Lee actually did highlight some of the very basic aspects of myeloma, the progression and so on, because we may have new members to this group. And Dr. Mauli has also given the very nice basic background to the immune cells and the immune system and how it actually helps to remove foreign cells in our body. And this leads very nicely into this talk on CAR T cells. Actually, I think most of you in the audience would have heard of CAR T cells. Anyone don't know what, or have not heard of the word CAR T? Okay, so one or two only, right? Why? Because in my clinic now, almost every day that I have clinic, the patients come in and then we go through talking about the treatment and so on. Then at the end they will say, Doc, what about this CAR T that I hear? Is this could I change to CAR T because I'm hearing that it's curing cancers and so on. So there is a lot of hype, there is a lot of publicity in the lay press as well, on the web, Google and internet. There's a lot of these information on the CAR T, which is why I thought that it would be useful for me to address this topic at this meeting and to highlight what are some of the truths and what are some of the potential myths so that we all could be on the same page. And there is no hype without some truth. So the idea is that CAR T was initially developed most successfully for childhood leukemia. Okay, so you see here a picture of children with leukemia. And why did it reach such high level of success is that using this technology, they show that even in children that has run out of treatment options, so usually these are kids that pretty much, is going to die within a few months because there's no other treatment for their leukemia, that some of them, not all, some of them, when given this CD19 CAR T cells that you see here, this word CD19 is mentioned in many of these papers, could actually be cured of the cancer. So going from a disease, you're not talking about just controlling or giving them another few years, they have no good options already, to a cure, is really something quite remarkable, okay, quite remarkable. But I would like to highlight that this is by no means for all the patients. So all the patients in the same group that has no good treatment to give, that has childhood leukemia, when you treat them, there is a percentage of them that may be cured, and in fact, the percentage is probably less than 50%. But this is quite a remarkable achievement because usually you wouldn't have anything that could be given for this. And of course, for every publicity campaign, you need to have a poster child. So in this case, it's truly a poster child. This is a kid, right? It is this girl down here that has, initially refractory cancer to now growing into a really beautiful young lady that is going to school, achieving well in school. Every year, in fact, she will put a card that is in front of her. She'll hold this card. This is my 19th birthday, 18th birthday. Every year, she's growing, right? So to show that this treatment is actually curing someone, right? Curing people. And so this received a lot of hype, a lot of publicity. This is like a big new hope in cancer treatment, and will be something that's useful for many patients, right? And so this word CAR T kind of stick in our kind of conscience, right? But CAR T just means a technology. So the CAR T that we use to treat leukemia will be different from the CAR T that we use to treat myeloma. So although the word is the same, just like chemotherapy, right, is one word. But the chemotherapy you use to treat myeloma will be very different from the chemotherapy you use to treat lung cancer, will be very different from the chemotherapy that you use to treat lymphoma, right? So every disease, although the same kind of technology or treatment can be used to treat the condition, it is actually different, okay? So although we are seeing that it's showing tremendous results and long-term benefit, because for the child, she has been disease-free for many, many years now, the same cannot be assumed for every single condition. So I think that is a very important starting point. Every disease is different, although the name may be the same. So I'm just going to go through some of these questions. What is CAR T treatment? Is it approved for myeloma? Just in case someone say that, hey, how come in China we can do this? Across the road in KL, I can go and get CAR T. So is this proper or not proper? I'm not saying anything about whether it's proper or not proper, but I'm just going to tell you that for myeloma later on, that what's the situation, right? Is it a cure for myeloma, just like what we hope to be, and what we saw in leukemia, childhood leukemia? And what are the potential side effects? So is it a safe, very easy, very safe procedure? And what does it entail? So I'll hopefully answer some of these questions and summarize at the end. So in Dr. Mauli's presentation and with the video, it's kind of introduced some of these players already. Antibodies, which are produced by the plasma cells, that actually will bind to protein on the foreign body, which in this case could be the cancer cells or could be bacteria, and it will eradicate the foreign body, right? So on here. And in fact, daratumumab, which many of you are receiving, is a kind of antibody that is manufactured and modified, so not natural from our body, but is manufactured, that actually serve the same function, targeting a protein that is present on the tumor cells called CD38, right? And then he also introduces these T cells that are actually immune cells that we have in our body that also recognizes protein that are expressed on the membrane of foreign body, but usually processed by the cells and usually associated with other immune proteins called like MHCs and so on, right? So it is more complicated because it requires the protein to be bound, but also additional signals and interaction between the T cells and the tumor cells. So it's a complex system, not so direct. Whereas antibodies are more direct. Once it binds, it brings in other cells, immune cells, to actually then eradicate the foreign body, right? So the body actually employs different system of immunity to remove foreign bodies. It's very effective. That's why we can live and in such a dirty environment. Actually, you see, we are exposed to bacteria, viruses, and so on all the time. The body actually have a very effective system which is very complex and employ different agents to actually eradicate this thing. So actually it's the same for cancer. Why is not everyone developing cancer all, at a young age and so on? It's because also we have an effective immune system to, by and large, for many people, to remove these early cancer cells. So they may detect 1 or 2 abnormal cells, they'll kill it off, right? So it's usually quite effective. But of course, as we age and with some interaction with environment, sometimes this system may fail. When you give it enough chance, sometimes it may fail. And when it fails, just like other important system in protecting our body, the cancer cell may start to grow. Okay, so why does it fail? Is that sometimes, as alluded to in the video, the tumor may actually develop some additional mechanism to shield itself and protect itself from these agents, So they go underground, right? They become invisible to the immune system. And the immune system, although they are circulating, they cannot detect them. Sometimes they develop additional proteins to kind of block this kind of interaction, right? So that although the T cells may recognize the abnormal protein, but these additional signals are unable to be activated because the tumor cells have, again, produced some other mechanism to block the additional signals. So the cancer cells are actually very, very clever. For a cancer cell to survive and cause trouble is actually not an easy thing, which is, again, why not everyone is walking around with a lot of cancer. So by the time the cancer can grow and develop and cause problem, it is actually already a very smart cell, a very smart group of cells. So our immune system have failed, really. So how do we then reharness this very powerful weapon, right? And that is where engineering has to come in, tissue engineering. And that's also the effort of development and technology that is being developed around the biomedical field, right? So we have to modify our immune system to make it active again, to make it effective again. And this is where tissue engineering come in, and so we can engineer these chimeric, which means a fusion. Chimera are actually a fusions of two different types of things. In the Greek mythology, it's some chimeric organism that has I cannot remember whether it's a lion head and some other body or something like that, but it's a chimera, right, made up of two different parts. So, so here we engineer chimeric antigen receptor. How? Because it has this part that recognizes the protein on the tumor cells that comes from the antibody, okay? But the antibody, when it's activated, actually employ quite weak killing immune cells, okay? So let's go back to here. It activates NK cells, monocytes, which are not very powerful killing cells, okay? Whereas T cells are actually very powerful, and they can have a memory, so you will always remember. If the cancer occur again, it will remember and it will kill it again, right? So it has this long-term memory. So it has these advantages. But the T cell system is not as good as the antibody system because it requires other signal. So it's a more complex system. So here we make the best of both worlds. The direct recognition of protein by the antibody part, link it to the T cell part that is very effective in killing the cell, so that now once I recognize and the protein on the cancer cells, I can activate like a T cell function to kill the cell. So it simplifies the process, make it more direct, and make it powerful, and potentially make it lifelong because it has a memory, right? So that is the chimeric antigen receptor, only possible because we can manufacture this and we can express this in the T cells and reactivate and make our T cells that has lost its potency or lost its ability to recognize the tumors in our body to become active again. So the advantage is it does not require the additional signal and is fast. Once it recognizes, it kills the cell. It doesn't require these kind of additional signals and so on. And so this is, for example, a tumor cells, and you see these T cells that are abutting. So what it will do is, in the body, this is what will happen. And with further understanding of the immune system, we can make further modifications to these receptors to make it even more potent, to somehow shut off the signal when it has too much side effects, so that it's not really always lifelong. And because if it's lifelong, once you have side effects, you may not be able to shut it down, and it can become dangerous. So we are able to build in these additional safeguards to either increase its potency or increase our ability to actually fine-tune, when it has side effects. So this is all the advance in the technology that's allowing us to do this. So now we have different generations of CAR T, but the ones that are in the market, approved, and tested very commonly in patients are these second generations CAR T. Okay, but I want to highlight that although there are CAR Ts, they are different, right? They are different signaling. The protein that they target could be different. So for example, the exact treatment that is used for the childhood leukemia may not be relevant to myeloma if that protein, CD19, is really not that useful for myeloma patients, right? So we need to find another protein that is useful for myeloma, right? The CAR T that's approved and used for childhood leukemia may have different signaling molecules inside, right? 41BB. And for the myeloma ones, the different ones in studies and trials, they could be different, right? So this may explain why the potency may be different and the side effects may be different. So again, if you read in a blog and say, oh, this one can cause serious problem going to ICU, brain problem and so on. If it's referring to the childhood leukemia one, it may not be the same side effects that we'll see in the myeloma study. So again, recognizing that the same treatment may be different, right? So how is it done? It's not straightforward like using a drug that you prescribe and a pharmacy give and then you just shoot it in. It's a bit more like your experience going through stem cell transplant, right? So we will need to collect your T cells first. So this is a bit like collecting your stem cells, the exact same experience and procedure. Then the T cells that are collected go into the lab. The cells that are collected go to the lab where we isolate the T cells, using some beads, right? And we activate them. And then we do this engineering. We need to transfer the chimeric antigen into the T cells. And usually this is done using deactivated viruses, okay? And the transduced or the modified T cells need to then be expanded because we need enough large volume to go back into the patient so that it can go and seek out the cancer cells and kill it. And then the reinfusion is exactly like the stem cell reinfusion giving back. So to the patient, the entire procedure is exactly the same as a stem cell transplant, right? You have cells harvested, and then after a while, a few weeks, you have cells infused. What is different is what goes on in the lab, right? The engineering and the manufacturing that goes on in the lab, right? So actually to you, it's the same, but actually what is different is what goes on in the lab. Is it a very simple, safe procedure? Is it as simple as a transplant? I would say that some of the side effects are definitely much more severe than the side effects that we see in the transplant because these are very, very potent treatment. And when you have activated immune cells, just like when you have a fever with an infection, it's because the immune system is active, right? You have fever, you feel achy, tired. So these are what we call cytokine release syndrome. But when you have a fever, it is a very toned down effect. Here we're infusing large number of these cells, and if they're really effective, you can imagine that you can be 10 times sicker than when you have a fever, right? And this can be severe, and it can affect the heart rate, the lungs, and so on. And some patients may have to go to ICU to be monitored closely and to have some additional treatment. So some patients can become very severe in terms of side effects. And because it's very effective in killing cancer cells, you can develop tumor lysis syndrome if you have still got quite a lot of disease. And usually for these treatment at the beginning is given for patients with relapsed disease who have quite a lot of disease. So the tumor lysis can be quite severe. And what happens here is that when the cells die, they release a lot of toxins that actually may damage your kidneys. Okay, so patients may go into renal failure as a result of tumor lysis, right? But all these things, as long as we know that they may happen and the clinical team monitor and recognize them and manage them, on the whole, it should be not a problem, okay? But you may still go through these ups and downs, feeling quite unwell before you get better. Some of the early treatment, particularly in the leukemia, these CAR-T can somehow go into the CNS, and cause some neurological effect, which again is different for the different construct that is used. Okay, so not a standard side effects, but some patients did develop it. But more importantly is a lot of these protein targets are not unique to the cancer cells. So they also exist in normal cells, like B cells and so on. So because the treatment is very effective, all your normal B cells and plasma cells will be also destroyed. And so you may have a long-term problem with your immunity, and you may need long-term replacement of immunoglobulins and so on. So you may have lifelong need to replace what has now become missing. And with that, your immunity should be okay, you should be able to fight infection and so on, but it's a lifelong commitment sometimes to having a monthly injection in a hospital and the cost of it that come with it. So it's by no means a very straightforward, simple treatment, okay? And really, the crux of it is to really identify the correct protein to target and then you open your whole world out to the different immunotherapy platform, of which CAR T is one of them. So for myeloma, what is the protein? And the protein is this molecule called BCMA that is expressed on plasma cells, which is the myeloma cells, as well as B cell, right? So we don't really go for CD19, which is used for ALL, because myeloma cells are different cells from the leukemic cells, right? And so in the literature, you'll read a lot on this molecule called BCMA, and that's really the protein that we are targeting now. But still, I think it's not optimal because it is not unique to myeloma only. It is also present in normal B cells, normal plasma cells. So you will get those cells affected as well. So the search actually is still on for a better protein target that may be more unique only to the cancer cells. Then next time our treatment will only affect the cancer cells without affecting the normal cells. So I say that although this is very exciting and we will see some BCMA immunotherapy approved in the next few years, I would also say that this is not the end of the story. This technology will continue to get better. The target will continue to get better as well. And to say that CAR T treatment in myeloma, people have gone for different targets, as you can see here. CD19, CD138, kappa. These early studies have all failed. Okay, so the word CAR T is the same. These are CAR T treatment as well. It doesn't mean that they will all work. Okay, so it's important to drill down to what type of CAR T. They failed, right? Until the group in the NIH did a study using CAR T that targets BCMA, which is this word here called B cell maturation antigen. You can see it at the top. BCMA, okay? So this is BCMA, CAR T, chimeric antigen receptor, T cells, right? And you see this remarkable response in some of the patients where before treatment you see so many areas of disease, right? You can see this on a PET scan. These are all areas of disease. 2 weeks after treatment, okay, 2 weeks is very fast. All gone, okay, all gone. And you see that your light chain kappa go down from a high level to almost nothing. So suggesting that this is very, very potent. But you look at the 12 patients you treat, right? You only see response in some. You see PR in the first patient, VGPR in one. This stringent CR1, which is probably the patient example that they show, is only one patient. Okay? So when we think about, is this the savior, we're going to cure myeloma, long way to go yet. Because you can see here, the response is probably about 30%. Okay? Since then, many other groups and companies have been developing this CAR T. So I show here 4 studies that are most mature, meaning that have recruited the most number of patients as most advanced. They all target BCMA, you can see here, and they have slightly different construct at the back end. The signaling molecules is slightly different. They use different ways to introduce the CAR T into the CAR construct into the T cells, but they're all virus-based. Some are retrovirus, lentivirus. Okay, but again, slight differences. And they all will need some type of treatment, just like your stem cell transplant, you need melphalan, right? For CAR T, they also need to give some sort of chemo to allow these T cells, when they go in, to be accepted by the body so that the body don't reject it, okay? And the dosing you see here is slightly different. Some are using lower doses, some are higher doses, and the schedule is also different. Some are given in 1 day, some are 2 days, right? So there's some variability. When we say most advanced and most patient treated, how many are there? This is 2017, so of course by now it's more. At that time, 6 patients in one trial, 24 patients in another study, 40 in another study, right? So by no means, compared to your daratumumab, your carfilzomib, your pomalidomide, which are treated in thousands of patients, these are very early treatment, right? These are mainly in very refractory patients with 7 lines, some of them 14 previous lines of treatment. So these are really very advanced patients. Most of them would have a transplant before. What is the response like? Overall response rate, 50%, 46%, 77%. The China study, that is now, this construct and product is now bought over by Johnson & Johnson. It's 100% response rate with quite a high percentage in strict CR, but you also see that this trial is the trial where the patients are more early on in their treatment, only 3 to 4 lines of prior treatment compared to these that are almost twice as advanced, right? So it could be that in the future we need to use this CAR T early on, that will have the effect. But the point to say is that although the response rate is good, we don't know whether they are cured or not, because these are 2017, that means the patients that has treated longest is probably only 3 years, right? So it's a far away to say that these people are cured. Suffice it to say that the treatment is very promising, okay? Because don't forget, these patients probably have no other treatment option already, but still are achieving this level of response. What are the kind of side effects that you see? The most worried are these things called CRS, which I said, cytokine release syndrome. The patient may go to ICU. And it's quite high percentage. You can see here 80-something%, but most of them are low grade, because the grade 3 CRS is actually very few. So this is good, because it means that we can manage this without much difficulty. Okay, so on the whole, the CAR T treatment in myeloma looks to be fairly safe, very effective, but the follow-up is still short. So we don't know whether it will be a cure or not. And these trials are still ongoing, so by no means this is approved by any of the agency, FDA, European Medical Agency is not approved yet. The treatment for leukemia and lymphoma, the CD19, is approved for treatment of leukemia and lymphoma. This one is not, okay, but we'll have to see. So the challenges that we face, I haven't even mentioned that, is the cost. It's the cost of the treatment itself. The leukemia treatment in the US is costing $500,000 one shot. Okay, but they say that it's a cure, so $500,000, you cure you rather than now you take daratumumab for 6-7 years, it's also $500,000, right? But not everyone is cured. So, so it's still a challenge. And don't forget, you may have lifelong commitment to having to pay for these monthly injection of immunoglobulins and so on. Those are ongoing costs, right? What is the best construct, the best target? Still not quite there yet. Different types of T cells, are some of the open questions. How to make it lifelong, because some people will lose its effectiveness after a number of years, and we still don't know what actually determine how to make it lifelong. How to control better the toxicity, the side effects. And also the engineering facility that's needed could be a challenge in some countries as well. So in conclusion, just some of the commonly asked questions, the FAQ, is this an exciting potential new treatment for myeloma? Yes, for sure. And I think in the coming years, we'll see even better and better product, better and better targets. So big hope for myeloma patients for sure. Is this approved for myeloma treatment now? No. No doesn't mean that you cannot have CAR T treatment, but you need to know that it's not approved. So when you go and have CAR T treatment in China or other places, make sure that it is in the context of a clinical trial. And when it's in the context of a clinical trial, these CAR T treatments should be free. Okay, so you don't go to somewhere in, I don't know, like Timbuktu, and they say, oh, we have a cure for this. We'll charge you 100,000, and it's already 80% cheaper than what you will pay for the US product, right? And then you go there and you think that it's a great thing. You pay 100,000 and it's actually not an approved drug. It's an experimental treatment, okay? Then there's nothing wrong with going for trials and experimental treatment because it's important aspects of getting new treatment and so on. But in those settings, the treatment must be free. There must be a consent form. They must clearly highlight to you what are the risks and potential benefit, and you must make an informed choice. It's not going to the market and buy a vegetable, that kind of transaction, okay? So I think that's what it means. Could this be a cure for myeloma? We don't know yet, because cure really does need longer-term follow-up. Is this treatment safe? Generally, yes, but there may be some serious side effects that is much more serious than the kind of side effects that myeloma patients are normally used to seeing, with our daratumumab, carfilzomib, Velcade, very different type of side effects. So you need to be aware of that, but they are, again, generally manageable if the doctors recognize them. What if I want to try this treatment now? Only available through clinical trial. Okay, by all means join. I personally have a patient from South Africa, interestingly, but live in Bali, so came to see me. And he said that I went to China for the legend, the last trial that I say, trial, right? and he went there and he had his cells already harvested. But he said that before I can get my cells back, they stopped the study, probably during the time that Johnson & Johnson was acquiring the company. So everything put on hold. So he said that I need, so he said that I need you to give me some treatment to kind of hold my disease, because and end up he used daratumumab. So J&J benefit both ways. But anyway, so daratumumab hold him, but it actually only give him a response that is a bit like PR. So he still have quite a lot of disease, but at least we're holding him. He's quite well. Then one day he come and say, hey, great, the China guys wrote to me again and say, I'm ready to go back for trial, right? So he went back for the trial, he got the CAR T, and his response was very good. He went into complete remission. So it's true, although sometimes we worry about what is going on in China, but he got the treatment and he went to CR, and he was very happy to share his experience with me. But this is all in the context of a properly supervised clinical trial with proper informed consent and so on, right? So I think I encourage people to think about this, particularly when you come to a stage of your treatment where the treatment options are becoming a bit limited. Do explore these possibilities. In my setting, I do know people that are doing trials in the US and in China. I can make referrals. But I would also say that these usually are hard to get in because there's a queue of people wanting to go into the trials. And the nature of the trials is that they are limited to certain numbers of people only. So it's not everyone who wants to go have. So there are some of these limitations. And certainly in Singapore, we're looking to opening some of our own trials so that our own patients can have access to these kind of treatment a bit easier rather than having to travel out of the country. So I think I probably, I was complaining that people are taking a lot of time. I probably took up too much time as well. So thank you very much for your attention. Yeah.